Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Bacterial and fungal preservation methods. Long-term storage techniques explained. Table of contents. Introduction to bacterial and fungal preservation methods. Agar slant cultures. Refrigeration. Paraffin method. Saline suspension. Cryopreservation. Lyophilisation. Freeze drying. Preservation at very low temperature. Preservation by drying in vacuum. Introduction to bacterial and fungal preservation methods. Biopreservation is the process of preserving the integrity and functionality of cells. Most bacteriological laboratories maintain stock cultures of microorganisms for educational, research, bioassay, industrial, or other purposes. A wide variety of techniques are available for the preservation of bacteria, and it may be difficult to choose a method for a particular strain that not only assures survival, but also ensures that the genotype and unique characteristics do not change. The primary aim of culture preservation is to maintain the organism alive, uncontaminated, and without variation or mutation, preserving the culture in a condition as close as possible to the original isolate. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Agar slant cultures. In microbiology laboratories, microorganisms are commonly preserved on agar slants. The slants are inoculated and incubated until sufficient microbial growth is observed. After incubation, the slants are overlaid with sterile mineral oil to a depth of approximately 1 centimeter above the slant surface. The cultures are incubated again for 24 hours or longer, and then stored in a refrigerator. These preserved cultures are periodically subcultured onto fresh media. During subculturing, a loopful of growth is taken. The loop is touched to the glass surface to remove excess oil and then used to inoculate fresh medium, while the original stock culture remains preserved. The frequency of these transfers depends on the origin and condition of the microbial growth. This method is simple, cost-effective, and allows bacteria and fungi to remain viable for several years at room temperature. Refrigeration. Pure cultures can be effectively stored at temperatures between 0 to 4 degrees Celsius using refrigerators or cold rooms. This preservation method is suitable for short-term storage, typically lasting 2 to 3 weeks for bacteria and 3 to 4 months for fungi. The microorganism's metabolic activities are significantly slowed down, but not entirely halted. As a result, slow microbial growth continues, gradually consuming nutrients and releasing waste products into the medium. Over time, this ongoing metabolic activity can lead to the eventual death of the microorganisms. Paraffin Method. The paraffin method is a simple and highly economical technique for preserving pure cultures of bacteria and fungi. In this method, sterile liquid paraffin is poured over the surface of the culture slant and stored in an upright position at room temperature. The paraffin layer creates anaerobic conditions and prevents the medium from drying out. These conditions help maintain the microorganisms in a dormant state, allowing the culture to remain viable for several years. Saline Suspension. High concentrations of sodium chloride commonly inhibit bacterial growth. In this method, bacteria are suspended in a 1% salt solution, which is a sublethal concentration, and placed in screw cap tubes to minimize evaporation. These tubes are stored at room temperature. When required, the bacteria are transferred from the saline suspension onto an Agar slant for revival and further use. Cryopreservation. Cryopreservation, which involves freezing in liquid nitrogen at -196 degrees Celsius, enables the long-term survival of pure cultures. In this method, microorganisms are rapidly frozen in liquid nitrogen at -196 degrees Celsius, with the addition of stabilizing agents like glycerol. These stabilizing agents prevent ice crystal formation, thereby enhancing cell survival during the freezing process. Lyophilisation, freeze drying. In the lyophilization method, the culture is rapidly frozen at very low temperatures, around -70 degrees Celsius, and then dehydrated under vacuum. This process dehydrates the microbial cells and halts their metabolic activities, causing the microbes to enter a dormant state while retaining their viability for years. Lyophilized or freeze-dried cultures are sealed and stored in the dark at 4 degrees Celsius in refrigerators. The freeze-drying method is the most commonly used technique by culture collection centers. The lyophilization process. In the lyophilization process, the microbial suspension is placed into small vials. A thin film of the culture is frozen onto the inside surface of the vial by rotating it in a mixture of dry ice, solid carbon dioxide, and alcohol or acetone at a temperature of -78 degrees Celsius. The vials are then connected to a high vacuum line, which dries the organism while it remains frozen. After drying, the ampules are sealed under vacuum using a small flame. These lyophilized cultures can be stored for several years at -40 degrees Celsius. This method is also used for preserving toxins, sera, enzymes, and other biological materials. To revive the microbial cultures, the vial is aseptically broken open, a suitable sterile medium is added, and after incubation, further transfers can be made. The process allows for the long-term maintenance of cultures without changes in their characteristics and significantly reduces the risk of contamination. Preservation at very low temperature. The organisms are suspended in nutrient broth containing 15% glycerol. The suspension is frozen and stored at -15 degrees Celsius to -30 degrees Celsius. Preservation by drying in vacuum. In this method, microorganisms are dried in a vacuum over calcium chloride. The dried cultures are then stored in a refrigerator for preservation. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!